now that we've talked a little bit about the actual cell as a whole, we're going to explore the boundary a little bit more. So really membranes. That's going to be kind of where we start with. A little later on, so not now, but a little bit later, like next unit, we'll talk about energy and enzymes. But for this lesson, we're going to look specifically at the membranes, that kind of barrier between the cell and the kind of outside world. Okay. So sort of plenty of molecules about what it looked like. Those are little water molecules on both sides, and this is the goofy structure that would make up our plasma membrane. So not really going to worry about enzymes or energy at the moment. We're just looking at membrane structure and function from this particular chapter. That's it. So we'll do the essential question easy enough here is, what is, I love this, the membrane structure and function? Okay. So being lazy, but it works, and it will be what we try to answer in the next couple of sections. So we call this thing a fluid mosaic model. That's how we describe the membrane structure. So it's kind of this patchwork of diverse protein molecules that are embedded in our phospholipid bilayer. So don't forget that unit 1, where we've talked about lipids or fats. So these are our phospholipids that are going to make up the bilayer of our membrane. It has some selective permeability. So that's kind of what we've talked about with maybe the, as silly as it is, for our little city model, it's kind of maybe our border patrol, you know, controversial, but we kind of remember what it's for, is for allowing things to enter and leave selectively. Not everything can go and leave, okay, or come in. The protein embedded in the membrane of the phospholipid bilayer perform a whole bunch of functions, and so we'll get to talk about those along the way. Okay, so this is sort of what we're talking about with that whole fluid mosaic. So notice just kind of the gray part and the yellow there, the gray and the gold maybe. That's just the phospholipids, as we have here. We're going to have other stuff in it, like cholesterols, which we talked a little bit about with the lipids and things like that. We've also got membrane proteins. All the proteins are usually, let me go back there. All the membrane proteins usually tend to end up being purple. So we've got lots of examples of purple proteins that are doing all kinds of stuff within the cell. And so notice that they're kind of embedded back here. There's more back here. It's just dotting the whole thing. We've got these guys up front. So that's why we call it a fluid mosaic. It's going to move, and it's going to be patchy like a mosaic. Okay. Here are some other examples that we could talk about: attachment proteins, receptor proteins, some junction proteins. We'll get there. So the phospholipids are really the key ingredient of biological membranes. So that's our major thing, is the phospholipids. And what's really cool is they kind of spontaneously self-assemble into simple little membranes. So the formation of membrane-enclosed collections of molecules was a critical step to the evolution of the first cells. So we could think about that, but I should have a video. You might have to remind me, but there should be a video, because they show kind of just putting phospholipids together, which have this kind of goofy structure. And you could just take the individual pieces and throw them into water, and they will form a structure where they kind of point to each other the whole time. It's crazy. I don't know why, but they just kind of spontaneously self-assemble. They just put themselves together that way. So here's kind of, not the video, but a picture that we'd look at. So there's water inside of it, and a bubble has kind of formed with phospholipids that they put into this water, which is fascinating. All right, so that's our basic structure. We'll look at it a little bit more, but what we want to get into is some passive transport across the membrane and not have any energy. So diffusion is the tendency of particles to spread out evenly in an available space. So particles move from an area of more concentrated particles to an area where they are less concentrated. Okay, so area of more concentration, or what we call kind of the higher concentration. Let's see if I can spell today. Higher concentration to an area where they are less concentrated. So we have down or lower concentration. Higher to lower. This higher to lower thing that we're talking about is what we call the concentration gradient. When we think we're moving from like a high place to, oops, my bad, a high place to a low place, notice we're going down the concentration gradient. So that's where that comes from. We're moving down the concentration gradient. Eventually the particles reach this kind of dynamic equilibrium. We say dynamic because it just continue to move. But eventually there won't be a net change in the concentration on either side of the membrane. Either. So here's kind of diffusion. So if we put in like maybe a dye or a food coloring on this side, and we've got a semi-permeable membrane here, what will happen is that these things will begin to diffuse across the membrane. These particles will move across. Some will even move this way. But they're just going to start moving and moving around. Eventually, we'll reach equilibrium where some will move this way and some will move this way, but overall, it's going to be equal on both sides. Okay? And so that's why it's dynamic, is because everything's still moving. So dynamic, we're talking about is it's still moving. Now that's not the precise exact definition of dynamic, don't freak out, but you know, I'm just telling you for this one, it's still moving. Dynamic is more like changing. So we can even see that here. So we start with our green ones on this side and our purples over here. And what they'll do is they'll move across. Purples will go this way. Some of the greens will go this way. And eventually we'll have about the same. So if you notice, there's about 6 of the greens over here, 3 of the purples. Over here we've got 1, 2, 3, 4, 5, 6 greens, 3 purples. So it's about the same, and they'll move back and forth constantly, but overall it's going to stay about 6 green, 3 purples on both sides. Okay. The diffusion across the cell membrane doesn't require energy. It's called passive transport. My favorite way to think about this, well, let's see, diffusion down the concentration gradient, does this all mean that oxygen enters your cells by carbon dioxide passes out of your cells? It's through passive transport. This doesn't require any energy, so think about it in people form. So let's say, for example, that we had to have this assembly, and oh Lord, so think about the beginning of the year assembly, but we didn't have the pack available. We had to go to the LGI, and we all had to cram all 400 students into the LGI. Okay, so then we say, oh, well, it's time to go. Do you think it's going to require any energy on my or any other teacher's parts to get you guys to get out and spread out and get as far away from each other as possible? No. Because you guys will be like, oh, get me away from these things. And that's exactly the way that molecules are working. They're trying to spread out. They're trying to get away from each other. Okay. Do a little thing. Look and see what happens to people during flex time. Do they all seem to move towards one area together, or do you notice that little pockets of it kind of spread out and they make kind of equilibrium in the flex area? That's an experiment. So we're going to come back to membranes selectivity. All these animations, I'm going to put them together and I'll make them available to you a little bit later just so you can maybe get a visual on what's going on. But they don't want to work while I'm in this particular mode, so we're going to just keep moving here. One of the most important substances that crosses by passive transport is water. And water, for some reason, because water is so special and cool, gets its own name. So when we have diffusion of water, it's called osmosis. Okay? So when people say, I try to get that information by osmosis, that is incorrect because only water can do osmosis. Okay? So again, the little water molecules, they can move sort of, so that's going to be osmosis. Again, another animation we'll look at later, and I'll make sure it's available to you, as always. If a membrane, permeable to water, but not to a solute, it separates two solutions with different concentrations of the solute. So remember, a solute is something that you dissolve in a solvent or in water. So this is the stuff in water. Okay, so water will cross the membrane because it's permeable to the membrane, and it'll move down its own concentration gradient until solute concentration on both sides is equal. Notice the solute concentration is equal. So you may have to come back to this part. We can look at it again, but here's an example. Like if we have this tube and we have water in it, and this particular thing is only permeable to water. If we have a higher concentration of solute, the water will typically try to go that route. It's trying to make an equal solute to water ratio. How I like to put it, if you look at these little pictures down here, is notice how friendly water is as a molecule. Like, it's going to take new stuff, and it's going to hug it, and it's going to be around it, and it's like, hello! Water is super friendly for a molecule. It's loving, it likes to hang out. So why, and it's equitable. So why would we just like hug all these, but then not let water hang out over here? So it's just mean. So what happens is that the water will move towards the area of stuff. So water will move toward the stuff. Okay, towards the stuff. Like towards the solute. Okay, because that's stuff and it's water molecules and they want to hug it and they want to love it. Water molecules might be like me if I go to Petco on adoption day. That's why I can't go, because I see all the puppies and I want to take all the puppies and hug all the puppies and take them home with me. I can't do it. I already got too many animals. That's water. It's like going to the stuff and seeing it and going like I want to love it all. So it's got to spread out equally. That's my best. If that visual does not work for you, figure out a new one. There's lots of ways that you can think about it. All right, tonicity. Actually, we're at 11 minutes. Before we talk about tonicity, this might be a good place to stop, take a little break, breathe a little bit, and maybe write a summary, write some questions out to the side, and then bring it on back. So tonicity is a term that describes the ability of the surrounding solution to cause a cell to gain or lose water. Okay, so its solution causes a cell to gain or lose water. Okay, so tonicity, or the tonicity of a solution, mainly depends on the concentration of solutes relative to the concentration of the solutes inside. So it's kind of outside versus inside. Inside on solute concentration. That didn't come out right. That's all right. Concentration. Okay, so that's what we're looking at here with tonicity. So what happens when animal cells are placed in various solutions? So an isotonic, by the way, iso is about, you can look up your prefixes on this one. Iso should mean the same. So this is telling us that the concentration of solute is the same on both sides of the membrane. So cell volume will not change. This means it's equal. So like if here's my membrane, here we'll make a little cell. There's my little cell. It's got a concentration of 4 things on the inside of the cell, and there's 4 things to the outside of the cell. You know, it's sort of evenly spaced. Something like that, okay? It's not exactly the number of things, but it's you get the point. All right, hypo. So hypo means below. Below. So the concentration is lower outside the cell. So what water molecules will do is move into the cell. Okay, so remember that means if the concentration is below outside, like maybe I have one thing, maybe I have like, I don't know, however many things that is in there. So obviously the concentration is higher in here and therefore lower outside the cell. So the water is going to go into the cell. Remember, it's moving towards the stuff. Remember, water moves toward the stuff. Okay, so it's going in. And what's that going to do to our cell? It makes it expand bigger and sometimes burst. There's my little bursting thing. Hyper. So hyper means kind of above or too much. Okay, think about when you're like, I'm hyper. That means you have too much energy, right? So the solute concentration is higher outside the cell, so water molecules will move out. So here's our cell. Maybe we have one thing in it. We have all this stuff on the outside. Where does water like to go? Towards the stuff. so it's going to go out, and that's where we get a nice, like, deflated cell. Okay, deflates. Okay, so that's kind of a weird one, but that's the basis for osmosis there. So if we move, get a little bit better thing here. So hypo is below, and all these tonicities are referring to the outside of the cell. So if the outside of the cell doesn't have a lot, but the inside has a whole lot going on, water will move in towards the stuff and it will burst and split. Plant cells are a little bit different though, because they like it this way, because they have that nice cell wall, so they'll just like push up against the cell wall. Okay, ISO is about the same, so that way we have equal concentrations inside and outside. For a plant, or for an animal, this is normal, this is good, this is ideal. For a plant, however, that makes it kind of wilted, like just kind of slightly wilted, where it's kind of just sad looking. Okay, in hypertonic, this means that we have above or too much, and that's going to be outside of the cell. They actually do this by the darkening of the watercolor, but I'm going to add little spots in here because that's how I do. So you can see the things. We have like maybe one inside. So the water is going to go towards the stuff, and that makes our cell shrivel. This is a whole thing for plants where it actually pulls the cell membrane off the cell wall. Really just screws up a plant entirely. It's what we call plasmolized. It pretty much is like cell death for a plant. It's like you can't recover that. Okay. So there you go. Those are what we look at there. And we'll get some animations, and if you're really not sure, just look up some YouTube videos. Okay, look at some things. All right, so for an animal cell to survive a hypotonic or hypertonic environment, we have to do osmoregulation. And that's control of water balance. It's not hard. Think of the word osmo, so osmosis, which we know is diffusion of water, H2O. And then regulation is regulating, so we're regulating the diffusion of water. That's control of water balance. Should make sense. Okay. Moving to the next one. So cell walls of plants, prokaryotes, and fungi make water balance issues somewhat different, because the cell wall of a plant exerts pressure that prevents the cell from taking in too much water. That's why you can, but you kind of can't overwater a plant. They'll deal with it. They like it. You got to water them a lot, okay? But it prevents them from bursting when placed in a hypotonic environment. So, but if you're in a hypertonic, it will both animal and plant cells will shrivel, and it will, not be good. Some doom. Okay, so that's what we were looking at one more time, just to see hypotonic levels is good. It's normal. It kind of makes a sturdy plant with the cell wall. When we don't, we've got kind of like a flaccid plant. Whenever we've got a really hypertonic solution, then we're shriveling up the cell wall, so it's peeling the cell membrane off of the cell wall. I'll get it out in a minute. Okay. So just a few things about transport across the membrane, and we'll talk about all of these things and work our way through them a little bit later, but we've got hydrophobic substances that easily diffuse across a membrane. Think about our little structure. Whoops. I'll build it for you again here. But think about our structure. If we didn't mention this, this is a hydrophilic head. So that gets along with water. And then this region is hydrophobic. So it's really easy for other things to kind of slip through there because it's going to get along with this area that's hydrophobic. Those are those hydrophobic little fatty tails. So hydrophobic things make it through the membrane quite easily. However, polar or charged substances can't get through that because that's hydrophilic, or hydrophobic, excuse me. I just realized phobic. There you go. All right. So charged or polar substances don't easily move across the membrane. Instead, they help with specific transport proteins called facilitated diffusion. So because it's still diffusion, it doesn't require any energy, and it does still rely on the concentration gradient. The only thing that we're going to do is we're going to have an extra facilitator, so a protein that will help make sure it can get across, like a little channel. So the best way to look at it is like a channel. So this is our solute, which is either kind of polar or charged in some way. Maybe it's an ion. And here's our protein. And so basically it just gives a channel so that way that particular solute can get through from a higher concentration to a lower concentration. Notice we've got, 2, 4, 6, 8, 10, and 2. So this is going to go from higher to lower. No energy. Okay. Some proteins function by becoming a hydrophilic tunnel, which is what we just looked at, the hydrophilic tunnel. So that was last slide. For passage of ions and or other molecules. Other proteins bind to their passenger, others change shape, some release the passenger into the other side. In any of the case, transport proteins help a specific substance diffuse across the membrane down its concentration gradient and thus require no input of energy. Okay, so those are some really key points there. All right. Take a quick break, and then we'll come back and we'll finish this one. All right, so because water is polar, diffusion through a membrane's hydrophobic interior is really slow. So rapid diffusion of water in and out of the cells is made possible by a protein channel called an aquaporin. So it's just a water pore. The creativity of the names, I tell you. All right. Basically, we're not going to worry about this one, but Peter Auger figured this out in 2003, and it helped us with some blood typing stuff, which was pretty cool. But we're no worries. We're not going to worry about that part either. Let's take it out. Okay, this one's a different one, though. This is a big thing. So all of the others go down the concentration gradient. However, we have active transport, and a cell has to expend energy to move a solute against or kind of up the concentration gradient. So that's a different one. It's worth noting. All right. Let's see. I'll wait till next one. So the energy molecule ATP supplies the energy for most active transport. Okay, so ATP. It's a little molecule we'll talk about later. But the following figure shows the 4 main stages of active transport just so we can sort of see it. Not animation. Okay, there we go. I'll put up the animations. But what happens is a solute will bind to a transport protein. So notice we've got binding and our purple protein we've attached to. Then we'll get ATP will provide energy to change the protein shape. Notice it opened this way and now it's opening this way. Okay, so ATP provides energy to change that shape. The protein will then return to its original shape once the ATP is done. And basically we'll just wait for another thing to come in there. So that's sort of the 4 main stages. But it's really 3. Oh, a little miscommunication between slides there. But that's how that's done. And if you notice, we've got 3 and 4 up here, and we're moving a 5th one up. So this is absolutely active transport. It requires energy in the form of ATP. Okay, we've got 2 other mechanisms to move large molecules across the membrane, because some things are a lot bigger than you can just push out with a little channel. Sometimes we have to get rid of bulky molecules. So there are 2 processes. One is called exocytosis. One is endocytosis. Again, the words are not all that creative. Okay, so exo is out, or like exit. Endo is in. Cyto means cell. And cis is kind of a process. So let's look at the word. So a process of things exiting the cell. A process of things entering the cell. So there you go. Process, Exiting the cell exports bulky molecules, and some of those may be proteins or polysaccharides. Process of entering the cell, we're taking in large molecules. Those are the words. Always break down the words. Let the words try to tell you what they mean. Actually, this should be a suffix. Whoops. Anyways, in both cases, materials can be transported in a package within a vesicle, a vehicle, as I like to call them, that fuses with the membrane. So our little cellular vehicle called a vesicle. So what that looks like, or we've got two types of endocytosis, two processes of entering the cell. One is called phagocytosis. This is the engulfment of a particle by a cell, wrapping the cell membrane around it, forming a vacuole. Phago means to eat. So this is a process of the cell eating. So engulfing a particle, we're going to eat it. Receptor-mediated endocytosis actually uses membrane receptors, so receptor-mediated for specific solutes. It only allows things to enter if they have specific receptors, specific thing that say it's able to receive this. And then it pinches in to form a vesicle, a little vehicle. A receptor-mediated endocytosis is used to take in cholesterol from the blood, for example. That's a good example. Okay, so we're almost done there, just to kind of show you. So this is phagocytosis. This is the cell eating. So notice we'll kind of get an extension of the cell membrane around a food particle. It will eventually kind of take it in and eat it, and it becomes a part of the cell. Receptor-mediated endocytosis. We've got we receive some kind of certain signals, and they'll tell us that it is okay to take this thing in, and it will make a little coated vesicle, which will go into the cell. So we'll have a separate coating. We didn't mention here, but exocytosis is really just the opposite. You'll see a vesicle like this move towards the outside of the cell. It will fuse with the membrane, and then all the junk will go out that way. So if we had exocytosis, that's what that would be. Just to show you. But there you go. That's the majority of it. So we're not going to talk about energy of the cell. We're going to quit here and find a good stopping point. It's a different section, so if you really have any questions or any, problems, just shoot me an email and we'll get it figured out.